Did you know that 4% of the audience here carry a genetic variation in their DNA that is very similar to that found in people with FSHD? That's 1 in 25 of us. As many as 1.3% of us have exactly the same genetic changes as those with FSHD, or about 1 in 70 of us. But I don't mean to alarm you. The chances of you personally having FSHD in this room are relatively low, although as many as 1 in 7,500 people in Australia will go on to suffer from FSHD. Aside, of course, from those amongst us tonight, such as Bill, who know this already. It is nevertheless one of the most common musculodegenerative disorders in the world and affects almost 1 million people. It is a muscular wasting disorder that typically appears in the teens and will continue to progress in fits and starts for a lifetime, affecting the face, arms, shoulders, stomach, the legs, and often leaving the sufferer in a wheelchair. In some cases, the disease also affects the blood vessels in the eyes. One variation of FSHD is the aggressive early childhood subtype, which is very severe and can leave sufferers cradle-bound. This was what our late patron and artist Monica Ellis suffered from, and ultimately the comorbidity of cancer associated with her illness tragically claimed her life this year. The pace of biological science has historically consisted of grinding hard work, punctuated by periods of great advances. Over 60 years ago, Nobel Prize winners Watson and Crick discovered the structure of DNA, the double helix, indicated in the top left-hand corner of this slide. The discovery paved the path forward for a deep understanding of human genetics. In the present day we now live in, biological science discovery is being run at warp speed. We can order our own personal genome sequenced over the web for a relatively modest fee. And if you wanted to, you could get more insights than you ever wanted to know about your relative genetic strengths and weaknesses. Unfortunately, though, nature has thrown us a curveball. On a scale of genetic and medical complexity from 1 to 10, FSHD is a 10. The majority of FSHD cases are caused by a shortening of the DNA near the tip of chromosome number 4 in what are known as D4Z4 repeats. As I mentioned before, some of us have this same feature, but fortunately this won't usually lead you to getting FSHD. This is because there are factors outside of your genes, what we call epigenetic factors, or in other words, environmental effects, which also influence the onset of the disease. The shortening of this chromosome then leads to altered expression of genes, which then cause the aberrant growth and ultimately death of muscle cells, which you can see in the photographs on this slide. Before I joined the Foundation, it was blessed to obtain the input from Professor John Rasko, AO, and he worked with Bill and a group of eminent scientists to initiate the first funding from the Foundation into discovery projects. These grants have enjoyed tremendous success, and the Foundation has been acknowledged to have played a significant role in enabling researchers to identify some of the critical genetic features of FSHD, including the involvement of genes with strange names such as DUX4 and SMCHD1. One measure of the success of these grants is the number of publications in top-ranked scientific journals. The Foundation funding has contributed to the realisation of over 20 publications by Australian and international scientists. Piece by piece, these discoveries, usually the result of 2 to 3 years of work each, have been unlocking the secrets of this insidious disease. We now know which genes destroy muscle cells in FSHD patients. We now know how this happens and at what point various genes are turned on and off in the development of the muscle cells, as illustrated by the colourful fluorescence microscope photo of FSHD-afflicted muscle cells on the cover of the scientific article in this slide. Genetic discovery marks the beginning of an exciting but arduous road into translational science, but I'm really pleased to be able to say that these genes are now being targeted for the development of therapeutics. To address the transition to therapeutics development, the Foundation has set up a strategy of providing research grants according to tenders which are awarded on a competitive basis. Our Therapeutics Grant Committee consists of 2 medical doctors, both of whom suffer from FSHD, as well as a neuromuscular physician and a biotechnologist. Our strategy initially was to bridge the gap between discovery and clinical trials. This has now shifted, and we find ourselves funding some of the first clinical trials of experimental therapies. This is indeed an exciting time. In collaboration with international researchers, we are funding projects which are screening thousands of compounds for their ability to modify the genes which are involved in FSHD. The next step will be to optimise these new molecules and test their safety as medicines. We are impacting the standards of care for patients now. We have sent Australian genetics laboratory staff to the world's best practice diagnostics lab in the Netherlands to be trained in the complex aspects of FSHD genetic diagnosis. Our vision here is to create a centre of excellence for diagnosis and set up Australia's first registry for this disease at a leading Australian research institute. We are running a bone fracture risk study. This study will, for the first time, measure just how much FSHD patients are at risk of bone fracture. Anecdotally, we know this to be true, and the impact on the quality of life of a major bone fracture can be devastating for someone who has compromised muscles. Such an event could end up worsening the progress of the dystrophy. This study involves an investigator in the United States and an investigator at Concord Hospital in Sydney. It's a true international partnership. The study is already recruiting patients, the results of which we can expect soon will be a world first. We have supported the involvement of clinical trial sites in Sydney and Melbourne to be involved in a global study of children with FSHD. The goal of this study is to understand the natural history and progression of the disease, and we need this information to design treatments. The Foundation is also preparing to set up a trial in human FSHD sufferers of a new biotech investigational medicine called follistatin. This is a copy of a human protein which we all have in our body, which normally functions to promote the growth and regeneration of muscle tissue. Whilst relatively non-specific to the mechanism of FSHD, we expect this protein may have the potential to help sufferers maintain or return their strength and function for everyday tasks, therefore improving their quality of life. There is an interesting backstory here in that nature has a way of throwing up scientific discoveries. The Belgian blue breed of cattle have a naturally occurring mutation in their gene for myostatin, protein which follistatin inhibits. These cattle are known as double-muscled for their extreme muscle bulk and lean meat, the opposite of that wagyu that you might order from one of Luke Mungan's restaurants. Such a genetic defect would never be tolerated in humans over thousands of millennia, as such muscle bulk would likely drive an intense need for calories in their diet. Ultimately, they'd die of starvation. However, there are very rare modern examples of families with genetic mutations in this pathway. These are real-life ex-men and women with astonishing strength from a young age, sometimes ending up as athletes. We do know that they are relatively healthy individuals, and that is reassuring from a safety perspective when contemplating a follistatin clinical trial. To summarise, the work of the Foundation is not simple, as we have provided over 30 grants to researchers in the past 6 years. We've helped researchers identify the genes and pathways involved in the mechanism of FSHD. We're now funding clinical trials that will impact the standard of care for patients. We're also on the cusp of funding new clinical trials of novel medicines. Clinical trials are not cheap, and to be effective here, we need to raise more funds. As is so often the case with scientific research, there are potential applications beyond just FSHD. Some of this research will likely turn out to be relevant to other muscular dystrophies, especially the proposed follistatin trial. And finally, there is tantalising recent research suggesting genetic overlap in the features of FSHD and cancer. So thank you for paying attention and bringing real hope to patients with muscular dystrophy. I hope you enjoy the rest of the evening.